Hi guys, this is Dr. Komal and welcome to KC Science Talks. Here we are going to discuss a question from CSIR Part B 2017 June paper. Guys, we are going to discuss this in detail. The question is metachromatic leukodystrophy is caused by a deficiency of aryl sulfatase A and affects the CNS. MLD is a lysosomal storage disorder, a disease due to dysfunctional mitochondria caused by loss of the myelin sheath, and fourth option caused by a defect in proteins of the nuclear envelope. So guys, the answer to this question is that MLD is a lysosomal storage disorder. So guys, what are lysosomal storage diseases? These are actually a group of hereditary disorders that disrupt lysosomal function. Okay. In other words, these disorders disrupt enzymes involved in cell metabolism, signaling, substrate processing, innate immunity, apoptosis, and other complex cell cycling processes. Okay. And because of the disruption in enzymes, this leads to accumulation of undigested or partially processed molecules, which leads to cell toxicity. Okay. Now, some more facts about this disease. Guys, in CSIR, they reframe questions. So that is why I'm giving you a detailed explanation about this, because next time they can ask you this question in a different way. So here are some more facts about this disease. Guys, metachromatic leukodystrophy is an autosomal recessive genetic disorder. In most cases, it is caused by deficient activity of aryl sulfatase A, which is a lysosomal enzyme and which degrades sulfated glycolipids. Okay. Due to deficient activity of aryl sulfatase A, sulfated glycolipids do not undergo degradation. Okay, they accumulate. It predominantly affects central nervous system, and accumulation of sulfatides leads to dysfunction and destruction of myelin sheaths in central nervous system and peripheral nervous system. Okay, and I have taken this information from research paper, so this is 100% authentic. So guys, that's all. I'll catch you with another question. Bye. Please like, share and subscribe.